There's Morgan, actually. Morgan?
Hello.
How's it going?
Good evening.
Yeah.
Afternoon, still-ish. Just reading this. Yeah. We were at like 3.5, right?
Yes.
Or that's
what's written down, at least.
Oh. Okay, good. Skinner, failed novelist. That was pretty funny in the Substack thing you pointed to.
That is funny.
I liked Walden too.
Yeah.
Okay. Sorry, I'm just getting things set up here. How's everyone? Hi.
Pretty good.
Good.
Yeah. You guys have seen the link to the Notion. I don't know if everyone wants to give updates for kind of, I don't know, how's your week's been or whatever. It's been a bit crazy for me, but it's okay.
My mom got COVID on Thanksgiving Day.
Yeah.
Oh, wow, yeah.
It made Thanksgiving really hard.
Yeah.
I believe it. This. Whoops. All right.
Has anybody read is it Eric Hull's book, like World?
The novel or the other book?
I assumed it was not a. It was some book about consciousness, the history of consciousness research.
No, it's interesting because I saw a book in the local bookshop. I think it's one of his novels, but I would love to see that book. I was actually going to mention. I think one of the links I was tempted to share somewhere. Yeah,
The World Behind the World.
No, I hadn't heard of that before.
Oh, you could be right. It says part of which covers the history of consciousness research. It could be that it's still a novel. Yeah. I was just gonna say, I mean, especially given.
I think that's the real one. I think that's his. I'll start sharing my screen so you can see what I'm saying.
Well, I was just gonna say with the Brown and Miller paper, like consciousness research will never have a winter as long as there's like research on anesthesia. Right? You know, like it can be very physiological.
Yeah, that seems right.
Yeah, like I don't know what everybody else is working on, but like, Brown's working on the anesthesia.
Right.
Yeah, and it can stay really physiological and like it's and Lord knows anesthesiologists need to know what they're doing. So it's really interesting from a neurotech standpoint, what they use to determine level of consciousness by anesthesiologists during surgery.
Right.
Oh, okay.
Okay, that one.
Okay. Mm-hmm. Mm-hmm.
So what is the description of it?
I'm checking out. Okay. Lord, it wants me to sign in.
Interesting.
So,
Oh, this is the one that was in the Slack channel, right?
mm-hmm.
Risk of another consciousness winter.
Mm.
I skimmed it, but I didn't read it.
Like those boom times.
Yeah, I was gonna say I don't think it quite maps to the AI summers and winters.
Again, like that PSW talk from like a couple weeks ago. It's interesting just from like looking at again like the history of anesthesiology and the history of what constitutes brain death or what constitutes death and meaning what constitutes brain death.
Yeah. Yeah, so this is the consciousness summers and winters. So let's see.
Anyway, I was trying to say, yeah, I mean, you guys saw what we did apparently, but the comments were interesting. I looked at this person's stuff, but they're kind of politically cranky, so I don't really like.
I like that description.
Yeah, and it's sort of like I'm an old-time cynic, unlike Republicans like Aristotle and Machiavelli, but I don't like Democrats that knows it all. And I was like, okay, it was interesting. There was interesting. I forgot. There was something that I looked that was interesting here, but it wasn't. This Mr. Science thing. It was the trail that led to this more interesting thing. Like he kind of critiques, science is XYZ, whatever.
Oh,
but this was I put this link in the chat too, which is not opening, but
so it's really the dictionary definition of Substack.
Yeah. Here we go. This actual article. I guess my update outside of the context of cognition features and stuff is that I think part of the project I'm working towards is maybe fall into the realm of like science and technology studies, which is the field I'm not that really familiar with, but seem to be brushing up upon anyways. And I'm a little bit curious if I could use some of the data set in this for one of the courses I'm in. I don't think I can because I think it's a little bit too not fit for what we're trying to cover. But basically this article is sort of about this article is kind of about as kind of pulled up this person pulled some quotes out of it, but basically saying like the machine of academic industries is making people be unoriginal, and it's very hard to do real research that challenges things. Kind of what you'd expect. But it was interesting to see. I kind of want to look more at this paper and the data that they're doing, that they found and how they're doing it.
What was the other thing that you showed, the blog post?
Which one?
The one that you just showed. Yeah, that one.
This was based off of that study. It's just someone commenting on that study. Which I went down this rabbit hole finding this guy's page and then this one, and then I was like, okay, what's something that's actually interesting? He posted this, and then he was quoting this guy. So it was this big extended comment chain basically for me. And the comments, this was made in 2022. It's July. But he's like, I've been thinking about this PNAS article from, last year on slowed canonical progress.
Yeah, I remember this.
And so on. So, I mean, I'm curious about stuff like this. The methods are this. I don't think I have direct access to this big of science raw data thing. There's some stuff that's on like a GitHub page. Like this looks like a really interesting data set. I might try to get access to it. I don't know if I can. We'll see. you could.
Well, I was just going to say that related to that, now this is, you're looking out, I mean, Web of Science is I think, even larger, but there was a literature review paper that Amanda and I, like, we were talking about, not the scoping, was it? No, it wasn't the scoping review, was it? Anyway, there was paper recently that was doing a lit review, and they were using a data set from PubMed that I wasn't familiar with. It sounds like they're using PubMed Central, or, you know, the fact that everybody has to archive their papers with PubMed since, you know, 20 something, gives you this really large text data set of science papers. And I just remember noting it as like, oh, you know, what's interesting about this paper is the references that they have for this text data set. Web of Science might be proprietary. I'm not sure.
There's for some other stuff I'm interested in for later on. I'm kind of curious about these things. I don't really know a whole lot about like science and technology studies. I mean, there's obviously, like, I know about a little bit about Kuhn and older people and that thing, but I mentioned I didn't. There's things up here that I didn't get to. Here it is. Like, I've been using a bunch of, you know, our friend ChatGTP lately, but this something about science and technology studies, allegedly, according to ChatGTP. You know, Bruno Latour is actually someone that the person I mentioned a while ago who was interested in something kind of about history of science. Mentioned Bruno Latour to me a few times. And also Donna Haraway also came up there too. So, like, I'm sure you guys know about these people, but I know of them, but not very much. So I'm kind of.
I didn't know Andrew Pickering though.
Yeah, he apparently is in that space. I think I asked about him in the chat. I have something else from below too. You know, kind of. This is pretty generic. We can go to publishing conferences. But yeah, okay.
I'm seeing Eric Hull's Wikipedia. I didn't realize he published with Tononi.
Oh yeah,
he did what?
This Substack person, Eric Hull, published with Tononi.
Oh yeah. I think that's how he did his postdoc or something.
I see. So it's definitely
Yeah, he's Eric's an interesting guy. I don't know him that well. I met him at a conference a long time ago, and someone's kind of bothering him about his views at the time, who I was friends with, and it was kind of awkward because he was being accosted on a stairwell about like his views, and like Eric was kind of like, yeah, like cybernetics kind of matters and patient stuff. What's that?
No, I think you're getting an echo.
Oh. What about cybernetics?
What's that?
What about cybernetics?
Eric Hull's talk was about cybernetics and like information theory, and it was pretty cool. But someone was like giving a really hard time about it, and it was just a funny memory. Avery was there, and also Angela. We were all sort of present at this. People who kind of passed through, not immediately active lab members, were at an event around that. So it's always kind of interesting how that comes up.
He's got a paper with Michael Levin too.
Oh, yeah.
I mean, that's just the trend. Everybody should have papers.
Odds are, yes, you will.
That's funny.
Did you ever meet Andrew Pickering, Bradley? I think he's at
No. I was reading about him. I think he was at UIEC until about 2007, and then he moved to England, and I think he's retired now. But
I was like immediately like, oh, this is a really cool set of things. But then I looked into his manual of practice, and I don't really know what to make of it yet. I don't know how I feel about it. This is all stuff for later. I'm not going to dive deeply into this, but just busy fun with Chat GTP, and then I'm like, oh, do this thing about like timelines and that's too crazy. This is obviously not real letters here, but timelines and walking down like corridors of time with trajectories, and it's like you're kind of far out GPT art. So anyway, I see Ant-Man wrote something there. Okay. Did you want to say anything about your update, Amanda?
Just that I was trying to get it recorded today and I didn't finish, but I'm almost done. So I'll record it and I'll share it tomorrow.
Okay.
And then I present it 7 a.m. Friday, or maybe 7:30.
Oh wow!
My session.
Yeah, your time.
Friday.
Right? Yeah.
Yeah.
Okay. I will see what I can do about being there, but I'm not going to promise I'm going to be there for that. I have some other things Friday morning I have to take care of too. Bradley or Morgan, I don't know if you want to share any updates or any comments, but you're welcome to do so before we start other things.
Well, just the point I don't know if you dropped off, but the point I was making earlier, just about consciousness research can't have a winter if there's anesthesia research to do.
Yeah.
Yeah, and again, I'm just really enjoying this paper. I mean, as Amanda's pointing out, like Hough and the techniques that they're using are well described in the by Brunton and et al. But I'm seeing a lot of really interesting overlaps in terms of this approach, and both the Oxford Dynamics package, what they're doing, as well as some other tensor-based techniques that seem to overlap with what's Hough, or this finite representation of Koopman operators, and it's really interesting.
Okay.
Yeah. But it's all about like trying to quantify, chaosity and nonlinear dynamics. But they're specifically using it as this measure of consciousness or loss of consciousness, or prerequisites of loss of consciousness. Yeah.
This is the one, right?
Yeah. So you'll see Emery Brown. I haven't. I don't know if they're together. They're both at MIT, so it's no surprise, I guess, that they're working together. But I haven't seen Brown. You see Brown presenting work on anesthesia all the time. But of course, Earl Miller's better known as like a neuroscientist, especially animal work. Cognitive neuroscience. So it's interesting. And then this particular approach is just nice because we're going to talk about this particular time delay embedding technique from Oxford for analyzing EEG MEG data that shares a lot of properties with this. Actually, they could probably both use like each other's some of the kind of when you dig into the method section of this, it's interesting how difficult the work is, as well as what you see in Oxford's approach. They're both using like multiple runs, and really have a lot of hyperparameters that are kind of data driven, let's say.
Well, we'll have to put that in a comment to a politically cranky fellow and Eric. Hey, there's other stuff happening.
Well,
Okay.
yeah.
Where did I go? Okay, so we go this way. So we covered all this stuff already. I don't know if you want to say anything about Amanda. This, so you put this here. We kind of this is just to say this. We just covered all this. I started unintentionally with like Eric and then that other person and the rant and the other paper, and then Amanda put this as a bullet point too, if you want to mention it.
Yeah, I shared that paper in the Slack. It was in the Human Use of Human Beings reading group page because it kind of talked about I don't think he used the word commodification of science, but that's kind of what he was describing, like the non-science forces that drive science. That seemed relevant to that paper you linked. And then, of course, Kuhn, who you already mentioned.
I was trying to sit in one spot, but I think I might have like bad acoustics, honestly. So let me try moving back here.
What is Bunny Book?
Well, glad that you asked, because at a well, I just lost my thing, but oh, I see. Are you still seeing the same image, like the same screen, by the way?
No, it's moving.
Okay, because I just plugged in something and it changed like monitors. So, let's see the best way to do that. Let's go this way. Man, I can't remember the name at all. The talking dog of TikTok is Bunny. I don't know if anybody's heard of Bunny. I am Bunny. That's the dog. Yes. Oh, well, okay, thanks. Bunny the dog is a sheepadoodle who displays behaviors of many human identifications. There's a bunch of buttons on the ground that say things like outside, hungry, angry, sad, poo, whatever. And like the dog apparently communicates with its owner. What I found out by the book and didn't know otherwise is that Bunny is currently subject of a series of studies by researchers at UC San Diego at the Comparative Cognition Lab. And in the book, it mentions that too. I did not get the book, but I saw the book today in the library. I didn't even think it was out yet. But it's really interesting to see, like if you follow them on social media, the dog and its owners, like you can see these, it's really interesting to see the stuff. And like I've kind of mentioned them a few times, or I mentioned like the stray cat analogy when my friend took in a stray cat, how the cat like changed its like kind of what it acclimated, and it changed its sort of response patterns over time. And it's kind of reminds me a little bit of this, but even though it's like directly associating sounds, like specific sounds like outside, good, etc. And so on. And it was interesting because number one, I guess part of why it's interesting is that like and in my classes today, this week, there was a really interesting discussion about we got into a subtopic on sentiment analysis. So like natural language processing related stuff. And it was all super basic, but the TA who was leading discussion was like, oh yeah, I'm working with this, and it related to some things I knew in like the health tech arena, but also I'm following up with them about it. But there's someone at UCSD is doing like an ADHD related sentiment analysis NLP kind of study, and I'm quite curious about it because I think a potential topical interest of mine in the future. Like I actually find sentiment analysis pretty interesting, and I'm seeing more and more avenues like for data science-y related stuff that I might actually want to do in the future, which is great because I wasn't quite sure like part of my approach to some of those things. But one area I'm particularly interested in is sort of like, the unpleasantly awkward and problematic nature of diagnosing like neurodivergent and or invisible illnesses, and like ways to push that envelope. I think are quite interesting to me, and I know several people, as probably many of us do. But I think I have an above average amount of people who are like severely affected by invisible illnesses in my life. So things that kind of affect accessibility or understanding of their situations, and like neurodiversity, especially about what is normal and so on, and the various factors at play are interesting topics. And I didn't get to talk too much to TA about what it was specifically, but I'm there's some interesting dialogues happening on that front. And hopefully more so in the future. And I might have more things to say about all that in the future. So, that's sort of the human version of it, and maybe the interspecies communication is another thing. And I do have a desire to merge some of our dear friend, Mr. Dr. Michael Levin, and his take on, diversifying things and the ethical social frameworks that comes in with that comparative analysis study. Also, Jenny from Plagiarism mentioned something about Dizzy again, so diverse intelligence that way. And I see there's a, the comparative cognition. Let's just look at that really quickly. I haven't looked at this before, so I'm a little blind with this, but there's definitely something in this space that I really want to get into down the road too, and I'm trying to. I think the best way to kind of come full circle and wrap this up to get back to the main paper is like, I really would like to, under the banner of the project I'm trying to develop into next year, I would really like to have a comparative cognition related theme in terms of how our understanding of that space affects maybe things under the umbrella of like science and technology studies or philosophy and history of science or whatever development progress buzzwords we want to throw at it. But there's definitely a something here. Dog Cognition Project. I'm sure that's like.
Oh yeah, I just dropped in some links that. So I don't know if you saw the thread I posted on bioevolution, but I got a bit of a dog thread there because I saw that there was this drug that had come out that was used for, they were saying it had significant effects in terms of longevity for large breed dogs.
Yeah.
And so that's already like fascinating. But it just spurred me to check on some other. The UCSF had a massive canine behavioral genetics project back in the kind of early 2000s, and I thought it was a brilliant idea at the time. Like, what a great kind of citizen science project that possible, just because pet owners are just so interested in their own pets, and it would be a great way to get lots of people to submit lots of data. And anyway, so I just kind of followed up on that. So you can see in the links a number of just interesting things that are happening in that space that NIH has followed up on that, and they've done a massive genome collection, and again, like trying to connect behavior with genetics. And so the what is it? The nature,
That was Scientific American there.
Yeah, the Scientific American just describing if you check the PubMed paper, it's I think, it's in Cell. Yeah. So the Cell paper on domestic dog lineages and behavioral diversification. So like to your point about designing intelligence tests. So this had come up in the discussion that we had previously about the need for different intelligence tests for animals, and being able to make them broad enough that you could get something that would work for, say, dogs in terms of finding different strengths that different dog breeds have, or different individual dogs have.
Yeah. Did you want to say anything, Bradley? Sorry.
Oh no, I was just interested in this, but
it's really weird string of like circumstances, because one of my engineering friends said to me earlier today, "Hey, my engineering job's cool, but I'm really enjoying data analysis. And by the way, I'm volunteering in an animal shelter, and I'm doing this massive data analysis project for people putting their pets and dogs into data shelters. And I want to do more in that space in the future." And it's like, okay, that's let's just move on to the paper at this point. We can spin around here forever, and that's fun. But I forget where exactly we were. I think we made pretty good progress, but also I think we're getting to like 2.3 or 2.4 or something. Let's look at last time. I think we put it at the. Why is it?
It was 3.5.
Okay, was it 3.5? Yeah. There we go. You're right. So 3 in general is neural codes constitute information about the world. 3.1 was coding or codes as information by reference. 3.2 is information as subjective laws or internal models. 3.3 is subjective physics of the Martian iguana, which is an interesting topic. Predictive coding and general models, Clark and others. And then we were just getting 3.5, which was can neural codes represent structure? I'll get this away. So yeah, I don't know if people want to give kind of where were we or their feelings about all that stuff. There are some really generic discussion questions here, but I think we can kind of skip them for later because we have plenty of things to talk about. Discussion systems. Okay, let me try to adjust this a little bit more. There we go. The previous paragraph was technical work on predictive coding is focused exclusively on the technical sense of prediction and coding. Therefore, no empirical evidence that such codes might allow organisms to perform predictions in a broader sense. Right? Can neural codes represent structure? This kind of representation, the words of the world, the kind of representation of the world useful for adaptive behavior is a structured internal model. Can neural codes possibly replicate that structure? That's what this chunk's about. Memories. The basic assumption of associative neural models of memory, retrieving memory consists of triggering activity in parts of the memory-specific cell assembly, or engram cells, which lead to the activation of all neurons in the assembly. Talks about problems with the cell assemblies. bag of words, bag of neurons. I think someone either called me a bag of neurons before, or remarked in a slightly humorously but derogatory fashion of someone else being a bag of neurons, which is bigger than other things, but still. Not only representing the lawful structure of the world, but also the structure of any given perceptual scene. Consider the example, the simple visual scene depicted in Figure 5, which is this one below. Try to zoom out a little bit so you can think about it here. There's Paul, a person I know, wearing a new shirt, driving a car in Figure 5. What is important here is that the scene is not simply a bag of objects. Objects have relationships with each other, and there are many possible different relationships. For example, there is a car. I'm trying to here. For example, there's a car and Paul has a specific relationship with the car, both physical, someone in the car, and functional, driving it. Some of my behavior depends on identifying these relationships because, for example, I can talk about them, and if behavior relies on neural codes, then those codes should represent relations, not just pixels of the image. But cell assemblies cannot represent these relations. Suppose there is a cell assembly that encodes Paul, another one, the car. To encode them, the driving relationship, the cell assembly that encodes driving would also be needed. But that assembly should also somehow refer to two assemblies representing Paul and the car. And this is something that cannot be done with an unstructured bag of neurons. So it's sort of like, because I've spent a lot of time there in this lab, your structured relations or your unstructured relations and your notion pages. Like, well, he's saying there needs to be some structure. He's saying you can't have that structure. I don't know how we feel about that. The binding problem. Does anybody want to say what the binding problem is? Since we're talking about this. Oops. I have an email.
Morgan knows. I mean, if he wants to take a shot at it. I could take a shot at it, but I just wanted to see.
Well, I was actually just going to point out that I have the bioarchive paper, and there's a figure missing from the actual published paper. So, but I guess it essentially it speaks to that in the sense that, like, when these different features are identified by different circuits, the binding problem is how to get all those features combined into a single percept. Would that be a
yeah, I mean, the binding problem was really big like 20 years ago or more. And the idea is that, like, you have these mechanisms that bind perception together, bind different types of perception.
Different types. And
yeah.
Specifically, Figure 5A, which is in the bioarchive paper, kind of speaks more to that.
Oh, okay, yeah. So there's this whole literature, and that's the reason I kind of asked Morgan.
Sorry. Go down. Sorry.
Is this it? This is 6 here.
Okay, switch it. Okay.
Number 6.
Okay.
Yeah. So go ahead.
The bioarchive paper is different.
Yeah. So yeah, the reason I think Morgan might have something to say about it is because there's this whole literature on, like, EEG signals and binding. So, like, there are certain like spectra of the EEG spectrum that are related to binding. I can't remember what it is, like 11 hertz or something. I don't know about that, but
you mean alpha being a kind of binding?
I think so. Yeah. I can't remember, but
it's like a binding beat or something.
Yeah. Like, there are large parts of the brain that all oscillate at the same frequency, and that's binding. And it's been contentious because they can never really find really good evidence of it. There have been a lot of papers published on it, but, like, it as a kind of a theory, it doesn't really. It's faddish, I guess, because you don't really hear about it much anymore.
Yeah. I mean, not a lot of. It does kind of remind me of people trying to make, like, illusions happen by timing things with alpha. Like finding someone's peak alpha and then trying to kind of either mess with that or present things so that you're not seeing them with, or you are seeing them synchronous with people's peak alpha. But it's hard to do, right? I mean, it's funny, like just on the LSL list, like it's still hard for people to get pictures to show up on the screen with kind of millisecond accuracy. So
right.
It's not surprising.
Yeah. So that figure 6, why don't we move down to that and see what that is? That looked interesting.
I'm gonna move this over here. Sorry, it's like.
But that's definitely like more like, you know, if things are happening simultaneously, and perhaps, or well, again, like you have different, you know, that your visual flow is broken down into kind of more particular or focused visual circuits, right?
Yeah.
And you know, the question then is like, how is that all? Why do we still have a kind of unified percept?
Right.
And you know, how is that all kind of come back together to give us that? And I like they're using a much simpler sound example here, at least to get it kind of, you know, well, this is, or is this more? Yeah, so they're not talking about trying to get spatial position, say, with hearing here. So I think this is just synchrony and what happens with neural synchrony, right?
It looks like in the paper they're going from like the binding problem, superposition catastrophe. One proposition is that the binding problem could be solved in direction of position objective label. However, objection does not address the broader point, which is that cell assemblies encode objects or features to be related, but not relations between them. In fact, it's known that current connectionist models, as we approach figure 6, which I'm trying to get to, or to design optimally to implement the idea features are represented by the activity of one or several cells, cannot be trained to detect very simple relations between shapes in an image. Binding problems led to several authors to postulate that synchrony to be used to bind features. But yeah, we're getting to figure 6 here. We want to talk about that.
Oh yeah, just that it's this idea that, you know, you have a source, an auditory source, and it hits the ears at different times, so there's a temporal gap between the two percepts. So left hand side, the right hand side. If it's skewed to the right, it's going to hit the right ear first, and so there's that intertemporal difference that you have, and the brain needs to like reconcile that so that you have a synchronized sound, so you don't have like an echo from one ear to the next, basically. And so this is what they're proposing, is that you have a source, it hits the ears differently. There's this temporal difference you see in the, I guess, in the spikes from A to B, and then that has to be somehow, you know, predicted or bound in some way that you don't have this, you know, you can detect things in space, you can localize them, but it's, you know, not a weird sort of effect or a sort of auditory effect. You do have auditory effects like that if you're listening to like a bullet ricochet around a canyon, you know.
So you are able to get a sort of 3D position, and it's interesting, you know, the studies that focus on that. Can you check the Ricci reference is also not in the bioRxiv version. I'm just curious what the Ricci L 2018 is.
This one?
Yeah, that reference there.
Awesome. Whoa. This one here? Same direction, different problems? And this one. Here, I think.
Interesting. Oh, interesting. So yeah, well, it'd be great if you don't mind dropping a link in the Notion of that.
Yeah.
But I was just going to say that since we mentioned Carl Prubram last week, you know, a lot of his work focused on visual system binding problem.
Yeah.
and so his languages of the brain, which focused on experimental paradoxes, and you know, it's certainly a lot of inferotemporal cortex, visual system, inferotemporal cortex, you know, seem to be involved in doing that. Unfortunately, from a brain imaging perspective, it's a really difficult area to image because it's right by the ears. Also, it's got some other problems. But his work would be good to go over in terms of experimental examples of the binding problem, and some of his theories about how to solve that.
Yeah, I would definitely be interested in reading some of that in this group.
Yeah.
So in this paper, the Brette paper, I don't know, his idea here is like there's some sort of synchrony or binding. Does he say that that's something that is needs to be a property of neural codes, or can be achieved without neural codes, or is impossible to do with neural codes? I'm not really sure.
Well,
go
ahead. It seems like he's saying that, yeah, in the part that he just highlighted. So maybe synchrony is an example of neurons encoding structure, but that's not enough. Yeah. I don't fully understand his point there that's highlighted, that it's only one type of relation. I guess I get that in this simple sound example, and it's a symmetrical one. I'm not sure what that means in this context. And then I don't see how, does Paul drive the car or does the car run over Paul? I don't see how that's supposed to be analogous to anything that he's been talking about.
I think that's to say the relationship isn't, you know, there needs a commentary on the relation for it to be appropriately meaningful, but this possibility remains speculative, and in particular remains to be demonstrated whether or not such hypothetical structures have the quality of representations that the brain can manipulate. So I don't really know what to make of that sentence, but basically it's saying skeptical and okay.
So now, really the brain is just representing like scenes or things, but it doesn't have any sort of structure to get us to causality. So you couldn't say like one thing causes another because you don't have that relational structure. So it's like, I can't say that necessarily, especially with like something like where you need to have causality to make sense of something. No, that's just my shot at it, but
yeah, I'm putting in a link as well.
Did you want to say anything about that link?
No, because it's honestly, I saw this blog post from Mehmet, and I was like, you know, I should really spend the time to fully understand this. So that's a note to self. He's talking about there should be no simultaneity rule in causal models, and I wonder if that's relevant to Brette's how that complements Brette's paper, 2012 paper in terms of neural synchrony. Was it computation through neural synchrony? Computing with neural synchrony is Brette 2012.
Oh, interesting.
Yeah.
Well, I guess this leads us to causal talk now. So the causal structure of the coding metaphor. The author gets in by saying, I've argued neural coding theory is general and representational sense of the metaphor. The idea neural codes are symbols standing for the properties that brain manipulates. But no evidence has been provided that this sense is valid. Worse, there's empirical evidence and theoretical arguments to the contrary, but here he focuses on a deeper problem in the metaphor. A striking character of this metaphor is a way to think about the brain independently of its causal structure. When we say neurons encode the location of sounds, we talk about the activity of the neurons without making any references of that activity or to the system which the neurons are a component. And he's going to get into this. Do we have any issues with this part before we go further? I think we've kind of talked about this a little bit before, but it kind of gets into his take on dualism and structure, causal structures here. Let's see. It is. He's saying the metaphor is dualistic because of how it's set up. Information that has been coded must at some point be decoded, etc. I think if people want to jump in and say anything about this, they can. I think we've kind of talked about these points a lot, but using the coding metaphor does not necessarily mean believing in dualism of the body and mind, but its dualistic structure has important consequences when it comes to understanding function. Dual components, encoding and decoding, are indistinguishable in behavior because no behavior involves just one of them. How is it possible to attribute function in neural codes? How is it possible to draw conclusions about the neural basis of behavior from properties of neural codes independently of the system in which the neurons are embedded? This is only possible by making additional assumption, namely that the encoding component has a function by itself, representing the inputs, somehow assigning the status of organ to a part of the nervous system. But there's no indication that the brain can be functionally decoupled in this way. Neuroanatomy rather seems to invalidate the hypothesis. Does that make sense to our panel? And I guess we're getting to figure 7 here. To illustrate this point, let's consider a concrete biological example. Paramecium?
Yeah, paramecium.
It's
the bag of neuron.
Yeah. Essentially, if you want to talk about this, Bradley, I think that's...
Oh, yeah. I think we covered this paper in Devo Worm at one point.
Yeah, I was going to say, this is like a separate paper that just came out, though, right? I mean, it was
last year,
recent.
I think, yeah.
Yeah, pretty recent, but really, really interesting.
I mean, this is like, where you have this non-neuronal cognition. You have a single cell that's doing things, and, it's able to do chemosensory stuff. So it's actually doing a lot of the things that a Bradenberg vehicle would do. So we talked about Bradenberg vehicles in last Saturday's meeting. We talked a little bit about that in the context of, these very simple behaviors. And, of course, Bradenberg ascribed human-like behaviors to them. But, if you just think about maybe C. elegans or maybe Bradenberg vehicles as chemosensory machines or chemosensory systems, they both kind of do similar things. C. elegans having a bigger connectome, because we were talking about C. elegans, the peptide connectomes, and so they basically they follow a gradient of chemicals. So if there's a stronger, if the source of the chemical is to the left, the paramecium swims there. It's attracted to sort of the increasing gradient of some chemical, and that governs the behavior. So it could be that, if there are two sources, it might kind of decide to go in one direction and then another. But, and so, C. elegans does this too with a larger circuit of neurons, by the way, because paramecium is a single cell. It's basically one neuron, although it's not a neuron. The cell is not a neuron. So that makes you wonder, why do you have, whole circuits for chemosensation if paramecium can do it with its own cell? It doesn't need any specialization for it. I don't know, but this is, again, his example of this non-neuronal single-cell cognition kind of going through the simplest system you can think of that would follow a chemical gradient. And now he gets into what the cell is doing, if it's encoding things, or if there's some sort of code that it's using to do that, or if it's just using some other set of mechanisms.
Yeah. Yeah. In this paper, just to show it briefly, is this a paper that was talked about before, or is this just Brette's?
This was we talked about this in DevoWorm last year. This paper here. So this is, and I think we did the preprint or something, but yeah.
But it's got an eLife paper too that came out before that.
That's it.
Yeah.
Excuse me.
Yeah, this is from this year, so this is actually a different paper, but it's fine because it's that same work.
Yeah.
But yeah, he's getting that's what he's getting at basically, is that you have the single cell, and I think with C. elegans you're getting chemosensation connected to different types of movements and other things, but in decision making, but still doesn't explain why you can do chemosensation in a single cell. Why do you need a whole circuit for it? Or why do you need big brains to do things bigger brains even to do things following chemical gradients or sensing light or whatever?
So I guess his conclusion for this section is, there's no indication that the brain is special, not special, in that it can be meaningfully separated into two dual components, which sensory motor system is arguably much simpler than the brain. Yeah. Okay. I don't know if we have any hot takes.
Well, in the meeting on Saturday, if you look through, actually the thumbnail on the YouTube channel has this, what did I call it? There's a model that I use, SIM. It's sensory, interneuron, motor. And it was the three neurons you would need to have a sensory motor loop in a system where you would have integration of different sensory neurons and then outputs to different motor outputs. So you have the three neuron model, which is a minimal model. And then, paramecium seems to be doing this in one cell. So we know from other examples of non-neuronal cognition that a lot of single cell systems where people would propose this might be using things structural biology or molecular biology to do this sort of work. And in fact, people have argued that there may be codes in molecular pathways or in structural structural mechanisms, in different parts of the membrane of the cell or other types of things that are computing. Basically, that's the argument. So that might be a stand-in for what we have with a bigger nervous system. But still, that's a contentious metaphor because, can you show that it's doing the same sorts of things that we expect from a code? I mean, the same criticisms apply that we're bringing up here apply to those kind of systems as well. So,
yeah. Coding versus causing. So in 4.2 it gets into, the man was writing here. The fact that neurocoding metaphor is a way to think about the brain that is disconnected from its causal structure, yet postulating that neural codes or representations we imply these codes have a causal structure of the brain. I think it's kind of the main discontent of his, one of them. The problem with neurocoding is defined as that of elucidating the transformations of informations in the nervous system. But does coding imply causing? The bold signal firing. I know people want to talk about like things like the firing rate versus spike timing debate. This is kind of interesting to think about in terms of like some of the cardio phenomenology stuff, I guess. Not. I don't really want to go there right now, I guess, but does the brain use the bold code, but versus like what things are influencing, let's see. Did you want to say anything about what you're writing around here, Amanda?
A little bit after where you have highlighted, talking about like how the heart works. He says, this means that the excitable cells of the heart encode running speed and their firing rate in the technical sense. I don't know. That just
about figure it, I guess, too. You're saying
if what's going on in the brain is analogous, then it I don't know.
Excuse me.
This paper has been it's been all over the place, and I haven't been able to spend as much time on it as I need to fully understand. But I guess I don't know. Through the whole paper, my intuitions have been like, I don't know, not sympathetic to the idea that neurons encode anything, because I just think that because the encoding implies some kind of decoding, that just seems like an extra layer that, in the spirit of like 4E cognitive science, you want to like reject those extra layers. But that's not a very charitable reading of this. And I think it's a product of my lack of understanding. But that bit about the heart kind of gets at what my intuition is like, yeah, there's a sense in which a spiking neuron is encoding something, but that's just such a weak sense of the word. I don't have anything more helpful to add.
Right. And I think what I was trying to get at with the cardiophenomenology stuff is like cardiophenomenology is kind of trying to get at like actual cardiovascular things affecting experience in a way to extend maybe sort of Varela's neurophenomenological aims to make it, to incorporate it. Earlier here, he was saying, oh, now we're making, these other organs part of the nervous system. And it's like, we just read a paper before saying, well, actually, yes, like the cognitive system, maybe does include sort of this aspect because of these components too. So it's kind of like depending on where you want to come from, you can kind of try to push it in either way. And I don't know how to feel about it yet either. Yeah.
Well, just wanted to also add that a lot of the techniques that are described in the Emery Brown and Earl Miller paper are taken from, originally were applied to heart dynamics, and trying to understand the heart dynamics and realizing that it was best understood as this nonlinear system. It also makes me think of something that we've emphasized, or I've certainly emphasized in terms of trying to explain why cerebral organoid, the kind of thing that Alison Mutry and Bradley Wojtek see in the electrophysiology of cerebral organoids, like if EEG is arising, if EEG like signals in sleep-wake states and neonatal rhythms, power law rhythms are emerging in cerebral organoids, that maybe we're just looking at physiology, and that what's that's not, doesn't mean that this thing in a dish is conscious. It actually means that what we're looking at is just that can arise so easily from kind of a bag of neurons. It's a structured bag of neurons. Our emphasis, at least in Bradley and I's discussions about cerebral organoids, that like you're not building a mini brain, you're actually just building a specific tissue type that is perhaps expressed in the brain, but certainly very artificial in a dish. And anyway.
No, I think that's a very interesting point.
Yeah.
I made a very...
So
go ahead.
They talk about, I forget what it's, dynamical systems with intermittent forces, or intermittent... I'm sorry. Let me just see here. Sorry, I'll look this up if somebody else has.
Yeah, I was just making my petty remark was about what about a bag of LLMs being manifestly consciousness, that some people are hyping up. Like apparently Sam Altman was fired and then rehired, but fired because of Q-Star, which somehow does middle school or elementary math problems and means it's consciousness now and stuff like that. So.
Best marketing work ever.
Yeah.
Like we're all talking about it.
The whole bag of like X is basically that it's not embodied in a system. Am I understanding that correctly?
Let's sort that out.
I did want to talk about the neural organoid thing because I think that's important. So neural organoids don't have experience other than being like these cells in a dish or in a bioreactor, but they still have action potentials or something. You can stimulate them. That's some experience, but that pattern stimulation is very unique to the organoid. So it doesn't have anything else from the outside world. So it's really kind of like a brain in a dish, and you're just kind of like, sometimes you stimulate it to, get it to maybe take one sort of fate decision or another and become a type of cell. But I guess you could call that experience, but that's fundamentally different from what we see in the brain. So, that means, we can still kind of recapitulate the complexity of a brain. We don't necessarily need experience, but then again, it's not really a brain. So it's hard to kind of nail down what exactly we're doing there. But I think, yeah, the coding part, we could generate codes from something like that doesn't have that kind of embodied experience. The question is it like something that's comparable to what you would see in a set of behaviors? And, what I'm talking about is like very simple behaviors like controlling a drone or a video game. And people have done experiments with this. They call them hybrids, where they have like a dish of neurons, and they have a little microarray that records the activity, and they stimulate it and record the activity. And then they like have these simple signals that can control something in the world. So people have done this with virtual environments, with things like drones or whatever. You just have to generate a signal, and it has to be a response to the signal that was, the signal from the thing that you're trying to control. So it's this closed loop feedback. So we can actually do stuff like that. And I don't know if you'd call that a code, or if you'd call that a response, or what. So I mean, there's something there that we're, these models of like organoids and hybrids. It's kind of like a gray area, I think, with respect to neural codes and things like that.
Yeah, and I think I might be taking what you're saying a little out of place, so if we were to put it back in line. But I think, and there's something to be said about, the space between, even if you could make an organoid, or if you're making organoids that do give off this, have a response and generate some kind of activity that we can recognize, like how, like, it'd be really interesting to kind of really look at like different levels of, I think, excuse me, my throat's messed up right now. Maybe a way to say this that maybe only Bradly will understand this because of the context. But remember, way back when I first joined the lab, my original idea.
Sorry, I need to take off for dinner here.
Okay.
So
okay.
I'm
glad you stopped by. Thank you, Morgan.
Yeah, thanks for stopping by.
Yeah, totally, really enjoyed it, and
yes,
yeah, we'll keep talking.
Awesome, thank you.
All right, talk to you.
Bye.
When you first joined the lab,
I had this sort of like toggling levels of like complexity or something like that, or levels of cognitive ability. I don't remember what I call it, but I guess what I'm saying here is more toggling levels of almost sort of inverting it to not either what will be around the organoid, or what will be around the agent, or will be around the brain through the environmental complexity, almost in a sense of kind of a critical period-ish sense of being able to isolate, okay, well, if this experiences, but it's never given the experience of this dynamic systemic interaction, or having to regulate something, either inputs and outputs from even a simple environment to a more complicated environment, what could you get at from that kind of a way of trying to peek a little bit around some like black boxy components? But it'd be, I don't know how to do that. I don't know if you can simulate an environment to that level of complexity and see what the thing you're trying to test would do in it to that degree. But yeah, I don't know.
Yeah, I think that's interesting that, you have this kind of, you do have these kind of levels of things where you have like we talked about the paramecium, we talked about the C. elegans circuit, and we talked about humans, and then organoids, hybrids, and they're doing different things. The neurons have, well, I guess, sometimes they have action potential. Sometimes it's debatable. C. elegans doesn't have true action potentials, but they have electrical activity, and they can communicate in circuits, or, even with the case of the paramecium, it's just kind of like some sort of thing going on that's coding of some type, or maybe like just some sort of information processing. So I guess you could, maybe have like that sort of idea of levels where, or like what Mike Levin talks about, competencies, where it's like, you have the one competency of just kind of single cell behavior, and then another competency of like a network that encodes like simple behaviors, but you can tune those simple behaviors. Then you have another one where there's a network where it can respond to the environment, that sort of stuff. So it's like it builds up from like a single cell to a network to a complex network to an embodied network to, whatever. And then it gives you different things. And there's a need for that sort of complexity. You don't just have it. But also, we have to remember that evolution sometimes does things in different ways. So some organisms have like a nerve net, some have a nervous system. Single cell organisms are doing things that other organisms need a network for. So it's not so much that it's, competencies like in a kind of a Scala-Natura type way. It's that, you have these different systems that can maybe represent things differently. They're not all related. It's like you have different pathways to getting to some sort of competency, but you can have these examples where you can see like, okay, well, simple systems do things a certain way, and then when you get to like a large network that's embodied, that you need some new processing tools or whatever, and so you can maybe pull it out that way and see what it looks like.
Yes. I realized I wasn't on camera for a long time. Hi, for a few moments left. Let me see. So, yeah, I want to go back. I was distracted for some of that, but I think what you said is really key and things that are on my mind a lot. And I'm going to process that for a while. So hopefully Mark will be able to join us next time. I think we're almost done with this here. We can maybe try to get to like the conclusion or something now. I do want to talk also about kind of next steps. We want a different next meeting. I'm fine with continuing this, at least to finish it, but then it's just there's a bunch of many replies. And I think we maybe talked about trying to scout them out to see, hey, does anything look good that you want to spend particular time on, as opposed to going through all of, like, I don't think we need to look at all of them, line by line. We can also ask many of the Chat GTP plugins to try to pull out interesting responses from it. I've tried some of the Chat GTP 4 plugins recently, and they're nice, but they're also a little. I think people would love them to be much better than they are. That makes sense. And I'm probably one of those people that would love them to be like, oh, type in and make a presentation off of a PowerPoint slide, and it's like the titles on like a slide deck, and it's like no formatting and no. So it's like, but you have a presentation now. It's like, cool. I think some of it's really good, but I digress. I guess we're at. We kind of talked about this? Do we want to talk about any of these figures? We mentioned the heart stuff. Figure 9 is in the section of causal power and coding variables. The causal structure of the brain. Also, I know Amanda just. Oh, you didn't know about it. Did you want to say anything about what you wrote here? Like closure and openness?
Oh yeah, when we were bag of neurons. My first thought was that what would separate a cerebral organoid, for example, from the brain is this, and then I think Bradley ended up saying this, that it's the brain is like a closed, not completely closed system, but it's like it has an organizational structure that the organoid would not have, and that made me think of just the way Varela talks about autopoiesis. And at one point, I think it was Varela, it might have been another author adjacent to him, talks about how operational closure is what like allows our openness to the world. It's what allows our experience as a subject, like in an environment.
Operational closure allows openness to the world, or freedom to say? Okay.
Yeah, like self-organization, inspiration from the environment is what allows our like interaction with the environment. And then I put a quote, but put it up where it should have been because it was earlier. I've already
This
forgotten
one
what
here
it was.
follows. Yeah. It follows that the efficient context-independent code does not match that one.
Yes.
No code is that adapted for the organism's goal. This mismatch occurs from the function between media. This also reminds me of, man, I wish if Dr. Johann John ever sees this, like, I miss your reading groups, and I would like. I can't do this without being tarnished by like American medical system and people having to make GoFundMe so they don't die. So it's like I would make a GoFundMe to help support a reading group from Johann, kind of a thing. Like, that's what I wanted to say, but it's like I just have such a bad taste in my mouth of people dying from that. Anyway, missing that because I feel like some of Johann's writing for a while he was really getting really close to this like metaphor and the deep primordial in and out divide between self and others, and like dipping into like some like cool like Taoist. I don't think he said that, but like my mind was going like, yeah, like these really like classical things coming up again. So that was yes. I think that's a super interesting topic and also one that's relevant to some other things. But here we are. We only have a few minutes left. I don't know if you want to try to do any of this stuff or just pick up next time, kind of ahead of the conclusion.
Let's just finish next time. I'll try to understand the paper we just read, and I'll look through the responses.
If you want any
good takes.
We don't really talk about anything with your paper presentation today, so if you want any other feedback or not, let us know.
Yeah.
That Google Doc that I shared is where I've been working on it. So there's everything I have is there. There's actually a draft of the talk now, and I'm about done.
Okay.
There's more on my mind. I'll just do teasers for other stuff. There's a lot more on my mind about, yes, the papers, the analogy paper that I have been working on. I don't think I'm going to be able to do, I don't think I'll be doing anything at the event this weekend. That's okay. I will still be pushing that stuff forward. There's an interesting, maybe you can see this here. I picked up this book today, Escape from Model Land, which it was on sale. But it's, there's a really interesting, there's a whole, I think there's a chapter or a section at least on models as metaphors, which is kind of relevant to that. It's by someone who's like Erica Thompson's LSE in London. A lot of mathematical modeling stuff. It knows about mathematical models, but there's an interesting overlap between my paper about analogy brain to mind stuff, and also I want to take what I'm learning in my courses so much into let's talk about like the philosophy stuff behind it, and I get opportunities now and then, but like the teacher was saying something about, oh yeah, well, a lot of these things are like a convergence of theorems that come together to make this result. We're using this result much easier, and it's like, okay, like let's start teasing it apart. And I know it's not really time for that to do it, so I'm trying to organize this and organize my thoughts on that. And I guess what I'll say in conclusion for this is like, I think the best framework for some of my bigger projects might be sort of this science and technology studies lens, but I'm so new to it, I don't really know, because I don't really want to just do. It's like there's a component that I feel like we're doing here in Cognition Futures ultimately is like what in like the 2 minutes we have left, like it's been a really interesting discovery for me to go from kind of this big, way before the lab, what happened in cybernetics, into okay, research and understanding this stuff, interdisciplinary all that, and then kind of framework vehicles and Varela and embodied mind, and how I originally met Amanda in that reading group where they raised stuff in the past. And now Cognition Futures has been like, okay, we're kind of looking at methods. That's our intent. And what we've really done is look a lot of like this sort of phenomenological pathway, and Varela's sort of work and the offsprings and the counterpoints to it. And what it's kind of, it's like a microcosm or like a specific case study of the bigger things, which is sort of about the role and nature of discussing sort of personal or normative experiences that aren't at least immediately empirical in the way that the science has happened in the last while, and it's like, what do we do with that? And I'm being very, very, very vague right now, but I'm trying to square all this into a more structured plan of action, because I think the core for me is basically what we're doing and getting really deep in the weeds here, but also there's a broader picture that I'm trying to suss out what to do in terms of both sort of it's kind of the same approach of the three things we're doing here in this group, like prescriptive and descriptive and then evaluatory. Like they're sort of the same thing at another level too, that I feel like is going on in the background of my mind, other minds, but like, okay, what do I do with them? So that was super nebulous, and I'm not going to explain it further because we're out of time. So I effectively use my time, so I don't have to explain it, which is great. But I'm just saying that kind of allude to some of the stuff that's going on there, and I will be kind of talking and messaging more about it over time. That said, next week, I don't think there's any major holidays next week, but next week, Wednesday is still good, and maybe the next Wednesday after that. Are people okay with that, or do you want to like stop? There will be a stopping for like some holiday stuff soon, I'm sure.
Yeah.
I'm good with next Wednesday.
Next Wednesday at least? Okay. We'll aim for that and go from there. Yeah. Finish this paper, and then we might have some open, maybe we'll have a little bit of, we can either think about something else we want to talk about, or like just maybe some step back and do some like actual admin discussion and think about how like Amanda's stuff goes and my thing did or didn't go, and what we want to do after ISPSM focus. Maybe something like that.
Yeah, I think we should have an admin meeting next week. I think it's time. So.
Okay. Well, hey, thank you so much. I'm really glad Morgan's been able to come to some of these too. It's a great experience and things that I always want to know more about. So thank you, everybody, and we'll see you all next time.
See you.
See ya.
Thanks.